We're going to kick off the meeting with our first presentation. It is entitled Inhibition of PANX1 Channels, Reduce the Malignant Properties of Human High-Risk Neuroblastoma, a Novel Target for Drug Repurposing. And I'd like to welcome Dr. Stephanie Langlois to the stage.
Most neuroblastoma patients present with high-risk disease for which better therapies are needed. When we started this project, not much was known about the role of PANX1 in neuroblastoma, except there was one study that has shown that N2A cells, which are mouse neuroblastoma cells, express PANX1. PANX1s are known to form ATP release channels that can be inhibited by carbenoxolone and probenecid. Interestingly, these two drugs are already used in clinic to treat ulcers and gout. And in that study using N2A cells, they have shown that probenecid can inhibit mouse neuroblastoma cell proliferation. So based on that, we're wondering whether targeting PANX1 could act as a potential new therapeutic approach for high-risk neuroblastoma. So first, we looked at available database and found that PANX1 is expressed in neuroblastoma of all stages. And when we used a tissue array in the lab, we found similar results looking at PANX1 protein levels. Interestingly, though, when we looked at high-risk neuroblastoma, we found that those with a higher PANX1 expression were having an overall decrease the survival probability. So to further look at the role of PANX1 in high-risk neuroblastoma, we obtained 7 patient-derived high-risk neuroblastoma cell lines, and as you can see here on the left, our Western blot data show that PANX1 is expressed in all these cell lines. So we then wanted to see whether PANX1 form active channels in these cells, so we perform a dye uptake assays, compare them to control negative cells which don't express PANXin, the HAK293 T cells. You can see that an increase of dye uptake incidence in all these cell lines, which was inhibited by probenecid, indicating that high-risk neuroblastoma express active PANX1. So to start assessing the role of PANX1 or how the effect of targeting PANX1 in these cells, we first treated the cells with probenecid for 48 hours, then submitted them to a cell proliferation assay. As you can see here, the black bars are the one treated with probenecid, the white bars are the one treated with the vehicle control, and all 7 cell lines, probenecid inhibited their proliferation. We've obtained similar results when the cells were treated with carbenoxolone. So interestingly, in 2015, there was one study that was screening neuroblastoma cell lines to look at proteins that were phosphorylated in these cells. And one of them was PANX1, and they've shown that this was phosphorylated on its tyrosine 10. However, the role of this modification was not known. So to start looking at it, we engineered a PANX1 mutant that could not be phosphorylated and called the YTNF. And as you can see in our channel activity assay, this mutant has reduced channel activity, and when expressed with wild-type PANX1, it inhibits its function, indicating that it behaves in a dominant negative manner. So based on that, we expressed this mutant in 4 of our neuroblastoma cell lines, and we've seen that it inhibits their cell proliferation, further strengthening the link between the inhibition of PANX1 channel activity and the subsequent inhibition of neuroblastoma cell proliferation. So based on that, we wanted, again, to go back to probenecid and carbenoxolone and further assess their effect on neuroblastoma malignant properties. So for this assay, we went through a 3D spheroid model, which, because 3D cell culture systems are thought to best mimic the in vivo situation. So we engineered these cells to express GFP, the green fluorescent protein, so we could actually measure the tumor size using our live imaging InQSight system. And cells were treated with probenecid or carbenoxolone at the time of cell seeding. So as you can see here with the black bar, cells form spheroid that grew quickly in the vehicle control. However, treatment with probenecid and carbenoxolone significantly inhibited tumor growth in vitro. We tested this with 2 other cell lines. We've seen similar effects. So for further translational purposes, we use a similar assay, but this time what we did is that we let the cells forming spheroid, which take about 48 to 72 hours depending on the cell lines, and then treated with probenecid and carbenoxolone. So again, the cells treated with the vehicle control, shown in black here, kept growing quickly. The one treated with probenecid showed a reduction of their growth, but interestingly, the one with carbenoxolone actually showed a regression of these in vitro tumors. And again, we tested this in 2 other cell lines. We've seen similar effects. So we're quite encouraged about that. So we then moved on to a mouse model. So in this model, we injected the askin B2 cell lines into the mice, treating them with either the saline, shown in black, probenecid in red, or carbenoxolone in blue, and then tumor volume were measured every 2 or 3 days. So as you can see, treatment with probenecid or carbenoxolone significantly reduced tumor growth, and we have also noticed an improvement of the survival. So again, we tested this in the other cell line, SHY-Y, similar effect, inhibition of tumor growth with probenecid and carbenoxolone. And we've also seen an improvement of survival. As you can see that some of the mice treated with probenecid or carbenoxolone were still alive at the end of our experimental period. So based on that, what we've shown is that PANX1 channels regulate high-risk neuroblastoma malignant properties through a process that involves its tyrosine 10 phosphorylation. Moreover, targeting PANX1 using probenecid or carbenoxolone may provide a new therapeutic approach for high-risk neuroblastoma. Thank you. And here are some of the people that worked on the project and some of the funding that made this possible. Thank you.
I'll now open for any questions from the audience.
Thank you. Erica Newman, University of Michigan. I really enjoyed this. As you know, neuroblastoma, particularly high-risk neuroblastoma, is a significant problem in our field. I have two questions. The first one is, did you know or do you know the sort of upstream regulators of PANX1 or downstream?
We don't know. We started to look a little bit to the EMT pathway because it's one pathway, the epithelial to mesenchymal transition that is known to be regulated by PANX1. We've started to look at things like beta-catenin, and what we've seen is that we don't see a difference in their levels, but it seems to going back to their normal localization when we target PANX1. So we think it might be a way, and PANX1 is known to often regulate differentiation as well. So we're looking into that, but that's the next step, and we're interested in trying to figure out which kinase phosphorylate PANX1 as well, because maybe if it's more specific to neuroblastoma, we can find, hopefully probenecid and carbenoxolone work well, but if it's too broad and we need to go a bit more into a targeted strategy, maybe that could be something too.
And then the second half of that is, did you notice any differences between cell lines that were MICAN amplified or non-MICAN amplified? And then lastly, you, can you talk a little bit about your mouse model? Where did you inject the cells and sort of how you, was this a metastatic model or not?
For the cells, the model, you mean?
Your animal model.
Yeah, so so actually one of these cell lines was MIC amplified, the other one wasn't. And the cells were injected into the flank of these immunodeficient mice. That's answering that question.
Yes.
Casper Wong, Los Angeles. Oh, sorry, please.
Jerry Hassey from Colorado. As someone who spent 30 years doing more and more advanced surgery, somewhat mutilative for high-risk disease, I'm very excited about what you and your colleagues from Ottawa have done. I've watched sort of intensive chemotherapy, then bone marrow transplant, immunotherapy, and now repurposing of drugs. I have one simple question. Are you going to now take this work to the next step, and do you have other classes of drugs that are FDA approved that might be repurposed for the same target?
Yeah, you're right. So one thing we would like to do, because right now we'll not be able to give that alone to the treatment, to the patients anyway, so it would have to be combined with, I guess, the regular chemotherapy drugs. So what we want to do too is that trying to combine them in the lab and see what happens would be great if we could actually reduce the overall concentration of drug and see if that could be beneficial. And one of the things is that probenecid and carbenoxolone were the first two that, PANX1s are pretty new in the field, and probenecid and carbenoxolone were kind of the first that were identified as clinic-ready drugs that target PANX1. But after that, there was spironolactone that has been identified as a PANX1 inhibitor as well. It's broadly used with kids and adults, so that's one drug we would like to try as well and see.
Thank you.
Casper Wong from Los Angeles. Great work.
Thank you.
Related to your introductory slide where you showed that expression levels are present throughout all the different stages of neuroblastoma and that high expressors, low expressors have different outcome. My question is, have you sequenced this gene amongst all of the neuroblastoma patients to be able to look at specifically polymorphisms that are related to gain of function, loss of function? Is it even that much more granular in terms of its functionality?
Yeah, no, it's a great idea. It's something we've done, and we are working out with our ethics board as well because we have the ethics approval to collect neuroblastoma, but also to isolate cells and being able to manipulate them and things like that. That may be something to incorporate as well with our own samples at SHIELD.
Thank you.
Thank you.
Thank you.